Hello and welcome to the Bench to Bedside e-colloquium in Lyme and tick-borne diseases. After recommended treatment for Lyme disease, the CDC notes that 10 to 20% of people remain with symptoms or relapse with symptoms. In the general literature, that prediction is 10 to 50%. And probably the most accurate studies show it to be about 36%. Dr. Lieger states very concisely in his introduction to his Antibiotics journal article, Repurposing Disulfiram, even with adequate antibiotic treatment, relapse of symptoms may occur. In some of those patients, we have proof that there's persistent infection. 2 of the 4 NIH randomized clinical trials showed improvement in patients when retreatment was applied, and some of them returned to full function in their lives. We are going to be discussing tick-borne disease today with 2 giants in the area. From our bedside, we have Dr. Monica Embers, who has a primate model of Lyme disease at Tulane. And from our bedside, patient side, we have Dr. Ken Lieger, who has been a clinical scientist and a master clinician for many years in this disease. Thank you, Dr. Lieger. Thank you, Dr. Embers, for being here.
For the introduction, and it's a pleasure to be here, and thank you for inviting us to talk.
So we are hoping that you two will discuss back and forth on various topics. And I'm going to give a short introduction that after recommended treatment for Lyme disease, the Center for Disease Control notes that 10 to 20% of patients remain symptomatic or relapse into symptoms. In the literature, the estimate is 10 to 50%. And I think probably the 2 best studies suggest it's about 1/3 of patients treated with the usual and customary treatment of 21 days of either doxycycline, amoxicillin, or azithromycin, remain or become ill. And that is Dr. Shattuck's paper from 1995 and Dr. Alcott's paper from 2013. So Dr. Lieger, can you begin by giving us some of the challenges and maybe a short history of the 40 years of Lyme disease in terms of the treatment dilemmas and the practices maybe that has led to over this time?
Well, I got into Lyme by pure happenstance. I didn't go looking for it. I just happened to open up an office in Armonk in Westchester County, which at the time was, I didn't know it at the time, but it was a burgeoning endemic or epidemic area. And I had a more general type of practice and just began seeing patients, again, like any internist would in a Lyme endemic area. So, and at that time, I would just follow the usual advice with fairly short courses of antibiotics, but it soon became very apparent to me that people would get better, and then in fairly short order, many of the patients would have relapse of the very same symptoms, and it was very puzzling. And just sort of intuitively, I would, or maybe it was 10 days the first time, they'd come back with relapse of symptoms, the same kind of symptoms they had originally. And sometimes even the rash, would recur, which is pretty unbelievable. So I would lengthen it, maybe double it, and then I'd end up tripling it, and then quadrupling it, and then for some of the patients, it seemed that whenever you stopped, their symptoms would come back. So you kind of got trapped into this open-ended antibiotic approach, which is far from ideal, but what else are we going to do for these people? So yeah, it's problematic.
Yeah, my first intro to Lyme disease was through a rheumatologist in San Francisco, Dr. Lavoie. And he would actually treat people, this was in 1989, with doxycycline or minocycline until they had no symptoms for a year. That was his feeling about the organism. And Dr. Embers, can you take us through the bench side of things? You started in 2003, but
Sure. What initially drew me into this field was learning about how Borrelia burgdorferi is able to evade host immunity. And basically, this is something that really interested me when I was a doctoral student. And I started reading, and basically Borrelia does all the tricks in the book to evade host immunity. And so we've learned more about that over time. But interestingly, the first step in identifying the causative agent of Lyme disease and starting to study it was the ability to culture the spirochetes. And you can't use regular bacterial culture media. It ended up being the addition of rabbit serum to the culture media that allowed the researchers to grow that out. And ever since then, we've been able to do host pathogen studies in mice, in dogs, in rabbits, and in non-human primates. And we've learned how difficult it is to manipulate the bacterium genetically, because most bacteria might have one or two extra chromosomal elements called plasmids. And Borrelia can have up to 21 different plasmids. And some of those are required for them to grow. And so it took a long time to figure out how to be able to manipulate the organism genetically so that we can knock out certain genes and learn their function. And so a lot of that work has come a long way. The understanding of arthritis and development in mice has been a heavy area of research, and we've learned a good bit about that. And we've also learned about how the bacteria can form antibiotic tolerant persisters in the presence of certain antibiotics. And so I think we've come a long way, but we still have a long way to go in answering the big questions that are affecting patients. We've learned a lot about the molecular pathogenesis and the host pathogen interactions. But now I think we're at a time where we really need to focus on what do we need to know about the bug that's going to help us help patients.
And knowing about the bug is important. Knowing about the bug in the body is important. So in terms of the ability to evade immunity, is that what Ken and I were kind of mired in when our patients kept coming back sick again with the same thing? I remember at the time thinking, well, you got to treat chronic sinusitis for at least 3 months. So maybe somehow we just have to extend this. But was it the ability to evade the host immunity? I noticed in your papers, you explained that antibiotics will reduce the pathogen load enough to let the immune system come in and do its job. That seems to be what you're doing with antibiotics. I sometimes think you're also revealing the bug. A dead bug can't evade the immune system. It can't manipulate it. So can I don't know, Ken, how you think about those things.
Well, over the years, we've learned through mostly the work of bench researchers the multiple ways in which the organism can, it's not just immune evasion, but also, sequestration, some of the early papers like Georgilis and Klempner with that model where they cultured Borrelia with fibroblasts, and with microscopy, they were able to demonstrate the Borrelia actually inside the fibroblast, and that it was immune from attack by antibiotics such as ceftriaxone. So that's one. Then, the work of Borson showing the transformation between spirochetal and cystic phase, and that the cystic phase seems to be impervious to the action of standard antibiotics. So there's a lot of different, I think it's become more comprehensible, and we've come to appreciate how clever this organism is. And of course, immune evasion itself is important, like the work that Nicole Baumgarth has done, which shows how the Borrelia kind of takes the germinal centers of lymph nodes in the spleen, and which usually have an architecture that allows that class switch from IgM to IgG. And by the time the Borrelia are done, it looks like the lymph node or the germinal centers have gone through a wearing blender, right? The architecture is all gone. And that also helps to understand why you can have these persisting IgM, antibodies. And like a lot of times us clinicians will see on a Western blot, not that much IgG, but an expanded pattern of IgM bands. And the conventional wisdom has been to ignore that, but it seems to me that that's actually revealing what's going on.
And I think
You're sorry. I think there are actually two facets of it. One in which the Borrelia are evading the host immune response and hiding from the immune cells, or growing very slowly and entering dormancy to where the antibiotics are ineffective. And then the other aspect where they're actively suppressing and subverting the immune response, which is what Dr. Lieger just alluded to, where germinal centers are supposed to form and they fall apart very quickly in Lyme disease. And so you don't get the generation of the adaptive immune response that you should get to a pathogen. And I think, one thing that makes this so difficult and controversial is that there's really no way to tell if a patient is recently infected. It's not like a urinary tract infection where you could reculture from the urine, or you could culture from blood of another infection. It's very hard to find these spirochetes, even though we know they're there. And the only way that we've been able to find them is to look at tissue sections in animal models to know, what kind of inflammation they're inducing and how long they're persisting.
Yeah, I mean, the only thing that we've had that's been practical and available to clinicians, but is fairly low yield, is PCR, in bodily fluids, and it's very hit or miss. If it's positive and if it's done in a reliable lab, the organism either is there or recently has been there, or its debris is there. And if you don't pick it up, you can't conclude that that's not there. And it's expensive to do that kind of testing, and the yield is not very high. So it's we certainly need better methods of direct detection. And I have this dream of some kind of a test that would be the equivalent of the viral load for HIV, that we'd have some measure of borrelial load, that we can have a semi-quantitative measure of the status of the infection in response to treatment interventions. That would be golden, would really help us. Right now, a lot of it is clinical, and that's difficult, clinical judgment.
I'm curious if in any of the animal models, there is an association between the load and the inflammation. Because I know both in your papers, in the early papers online, there was comments about how few spirochetes there were for how much inflammation you were seeing in the tissues.
That's an excellent question. And it's something that's very perplexing, because when you look at the mouse model, the the C3H mouse is particularly susceptible to infection with Borrelia burgdorferi. And when that mouse becomes infected, there is inflammation, there is arthritis, but it goes away. And we know that the antibody response comes up and reduces the spirochete load. If you look at persistent infection in non-human primates, which are incidental hosts versus reservoir hosts, you see a low-level infection, whether they've been treated with antibiotics or not. It's very hard to find the spirochetes. They're in different tissues. They might be in the heart of one animal and in the brain of another animal. And the inflammation, when we've done our studies, we use the histopathology as a roadmap to look for the spirochetes because, you know, there's so much tissue to look at. You know, we ended up looking at maybe 400 different slides under the microscope to find spirochetes. And there wasn't really a relation between the presence of the spirochetes and the presence of inflammation. So we know now that there are, you know, the spirochetes replicate, they release factors that can induce inflammation, like their peptidoglycan. And so what we think is that you don't necessarily need to have them in that particular location to induce the inflammation. And it could be inducing a chronic inflammatory response, a cascade in other areas, which is troubling.
And there's no prediction so far. You can't figure out why it's in the brain in one animal and the joints in another. And you're using the same strain. Out here in the wild, I often think, oh, well, if they come from the Northeast, they're more apt to have arthritis. And if they come from my neck of the woods in California, I see more neuro. But that's pretty loose, right? So you're using the same strain and seeing differences. You might see arthritis, you might see.
Yeah, rhesus macaques are just as genetically diverse as humans, maybe even a little more. And so we do see a broad array of manifestations, different tissues affected, and, you know, different immune responses to the infection, which is, you know, runs the gamut of what you see in patients. So we have a lot more to do. It's neat to have this model, but what would be ideal is if we could have some sort of biomarker that would indicate persistent infection or inflammation resulting from, you know, an induction of chronic inflammation in the absence of live organisms. We don't know, you know, if all the organisms that are lingering are actually viable and or dormant or actively dividing. My sense is that they probably are viable, but maybe in a state of dormancy for a period of time. If you look at syphilis, for example, that comes back years later, and we don't know where the treponemes are hiding out for that period of time between secondary syphilis and tertiary syphilis. So it's, you know, spirochetes have a really good ability to hide and to enter a dormant phase, and then, you know, something causes them to regrow or to slowly cause damage, and it makes them very cryptic and stealth. I know you like that word, stealth pathogens.
Very stealthy. Well, when the infection occurs in your monkey or occurs in humans, we know that they're persister cells, at least in vitro, if we throw doxycycline in there, or ceftriaxone in there. Is there, and I know that Ying Zhang in his Yin Yang paper, basically, if I understood it, said there's an equilibrium between the actively growing spirochete and the persister cell, and that there's always going to be some equilibrium. do you find that's true? Do we know? Do spirochetes automatically go into a dormant state in some places in the body after infecting?
That's a great question. We don't really know. We don't really know why spirochetes go to the heart of one animal and to the brain of another animal. We don't know why some patients have carditis and some patients have arthritis and other patients have neuroborreliosis. We know something about the pathogens and their virulence and different RST types that invade different tissues, but we don't know what signals are used to get into those tissues and start to cause damage. So that's a really good question, and it's definitely an area for further research.
I think,
as Chris was indicating, out there in the world and in nature, there's a diversity of strains even in a single geographic region. So you throw that in and it gets complicated.
and is there, you said when spirochetes are replicating, they can induce an inflammatory response by toxin, is it an LPS toxin?
So there was a recent paper out of the Jutras lab, which indicated that the peptidoglycan, which is part of the cell wall of the spirochetes, can on its own induce inflammation. And it was actually found in the joints of Lyme arthritis patients. And then they took that peptidoglycan that the spirochetes make and injected it into mice and were able to induce arthritis in the mice just with that component of the bacteria. And Borrelia have a very unique peptidoglycan. It's not like other bacteria. Again, a unique pathogen. And so maybe there are other components too that are doing the same thing. And we know that when the spirochetes replicate, they release peptidoglycan into the extracellular milieu. And so that can certainly be one mechanism by which the spirochetes are directly inducing inflammation.
In your paper on phenotype in regrowth, I was wondering if this is a lesson for us in giving our antibiotics. When those Borrelia regrow, they had a different expression of antigen, correct, than the ones that didn't regrow, for instance. And is there a concern that the regrown Borrelia may then not be susceptible to whatever you used?
That's a great question. And we did test that experimentally. So we grew the spirochetes in the presence of doxycycline. And when they regrew, we took that population and retreated it with doxycycline to see if they would be more tolerant of the antibiotics. And what we found is that they weren't. So whatever mechanisms are involved in antibiotic tolerance are not passed on to the next generation. So they're basically stochastic or random. So every population has the same number of spirochetes that may grow out to become persisters. And then as they grow, the same thing will happen, which is good. But also it's better than antibiotic resistance, right? But you have to figure out a way to kill those persisters as well. And so this is the beauty of the work that Ying Zhang has done, and in taking lessons from the TB field, where you treat with multiple different antibiotics with different mechanisms to try to get the growing bacteria, the stationary bacteria, and even the completely dormant bacteria or persisters.
And I noticed also one of your papers There's the spirochetes, the persisters, and the cysts. There are cyst forms, cystic forms.
We've definitely seen them. Dr. Zhang believed that they were infectious. He believes, I mean, he has some interesting hypotheses that have yet to be really tested, but are very unique, like the yin-yang hypothesis. One is that the tick has cystic forms in it, and those are the ones that end up becoming we don't really necessarily, we do see all forms in ticks and in culture, and we've seen round body forms in tissues as well. So the jury's still out on what the phenotype really is of these cysts or cell wall deficient forms. They've been called different things, but whether or not they're more tolerant of antibiotics, we don't necessarily know.
Well, and I thought, Ken, early on, and I don't think it was Burson and Burson, but there were maybe EM micrographs of a cyst with many Borrelia in them.
I'm not sure about that. I mean, in the Borson work, it showed transformation between spirochetal and cystic phase. I'm not sure about what you're referring to.
Okay.
But I mean, a lot of clinicians have attempted to use agents that in the test tube supposedly disrupt the cysts, but I'm not aware of any studies that have been done that actually document that there's a change in outcome. That outcome is better if you do that. It seems logical that it would be, but there hasn't been any really scientific study to answer that question. But even though it's commonly done, you know, like tinidazole and metronidazole is often used, and plaquenil, according to Borson, is supposed to also disrupt the cysts. But that would be a good area for investigation or formal study.
So another question I have to bounce off Monica back to Ken is, it seems like the macaque model would most closely imitate human infection. And I know we've talked about that some. And one of the things, of course, we worry about in humans is neurocognitive, neuropsychiatric, as well as fatigue, etc. So what's the clinical observation of your infected monkeys?
This is very difficult to assess in non-human primates. I think, you know, there could be decreases in activity level. There can be cognitive impairment, which we can test. We haven't done that yet. Most of the research on neuro-Lyme or neuroborreliosis in the rhesus macaque has focused on how inflammatory mediators in the brain and how that damages the cell types of the brain. So what does Borrelia do if it gets into the central nervous system to damage neurons? And we know that it does very effectively induce inflammatory mediators produced by microglial cells and oligodendrocytes, which are supposed to protect the brain. And so I think it would be nice to take this model a little bit further and to look at brain biochemistry and how you know, damaging toxins like quinolinic acid are produced in the presence of a natural Borrelia infection, and how that impacts their behavior and their, you know, we can do these cognitive tests to determine what the impact is. I think one of the more interesting infectious disease patterns is that of pandas or pans, where you have these children who have an infection with, you know, a standard, you know, strep throat infection, streptococcal infection, and almost overnight developed neuropsychiatric illness. And, you know, what is it that the bug is doing to the brain biochemically to be able to induce that? And I think that Borrelia and Bartonella both have the ability to do that over time. And we need to figure out what pathways are involved in generating that neuropsychiatric damage biochemically and at the cellular level to try to better understand how we can prevent it from happening.
In terms of the bug protecting itself, I think we've talked pathophys. What about biofilms? Can we touch on? I don't know. Do you approach biofilms in your mind or not really?
Well, I think there's reasonable data that it can exist with Borrelia, and according to the literature, organisms that exist within biofilm are highly resistant to antibiotics, like 1,000-fold or so, and that may be one of the reservoirs that, you know, Borrelia and other organisms, you know, reach out from after being treated, so it seems to me that the main thing to do to resolve the biofilm, if it is real, is to attack the organisms which create the biofilm, and then you have to have treatment approaches that can penetrate the biofilm and have a lethal effect on the resident organisms. So you know, I'm not a bench researcher, but it seems that there's credible work that supports that, and that may explain why antibiotics, which generally are fairly large molecules, are not really able to get at resident organisms.
So, you know, we know that Borrelia can form aggregates, and we know that the spirochetes can obtain a dormant or antibiotic tolerant phenotype. A lot of the work that's been done has been done in vitro, and it's very difficult to study biofilms in vivo. Typically, biofilms are multi-organismal and coated in like a glycolipid or a glycolytic matrix. Let me say that again. Typically, biofilms are multi-organismal, and they're covered in a sugar-based matrix. And so having the right tools to look at biofilms in vivo is really important. We don't have the best data to indicate that Borrelia itself is actively forming biofilms inside the human host. But there is some aggregation, there are some different forms. And you know, one of the aspects that I find most interesting is that when we look at antibiotics and how effective they are against any bacterium, we do the tests in vitro. We do it in a test tube. And that's not the natural phenotype of the bacteria. So, you know, one of the emphases in my lab has been to try to mimic the host environment and look at the impact of host adaptation and immune adaptation on the ability of the antibiotics to work. And so if there's some way that we can induce biofilm formation and test different drugs against them, I think that will be beneficial in the long run We haven't seen them in our non-human primate model. We've only seen, when we find spirochetes, they're single and they're rare, and we just haven't seen what I would call a biofilm. But also we don't have the antibodies or the reagents to look for whatever matrix the Borrelia are encased in. Some studies have shown alginate encasement, but Borrelia doesn't make alginate, so there have to be other organisms involved in that.
And I know a biofilm is made partially from the organism and partially from the host cell. I know in reading just a basic biofilm that, as you say, it's usually multi-organism, and there are multiple factors contributing in. One of the things I've always wondered is, since 98.5% of bacteria in the world live in a biofilm, they are sessile, they are not planktonic, that if you have a chronic infection, surely it will be figuring out a way to live in community, since it seems like everything does that. And whether that is a biofilm or is some other aggregate, as you say, I don't know. And obviously we need much more research.
Because anything, you need to have the right tools, and apply the right tools and the right methods to interrogate what you're looking for.
Yeah, and you almost, as you say, you need a biofilm growing in your test tube. You need somehow to create that organism ex vivo. If we look a little more at treatment, I know when probably early 90s, when I started getting more patients, and treatment changed at that point. So the ID docs were not treating with longer term antibiotic. I explored a lot via many of the doctors I listened to, Berscano et al., about how long you had to treat given the replication slowness of the spirochete, and how long you had to have that MBC or MIC for how many hours, and could you get it into the central nervous system. And I think this spawned, at least for me and colleagues, some really long treatments. I mean, my patient who got well after getting her Lyme at MIT and really being disabled by it, it took a year of IV antibiotic before she was back at grad school. So I just wonder what we've learned. I know there's now some pulse treating, et cetera. And I know you worked a little bit with that, Monica, in your treatment study. So I'm curious. Oh, one other, an infectious disease doc. I thought of the dormant bacteria as kind of in suspended animation, that they're just going to be there forever. And she said, no, they have to eat also. So just slowly, they eat very slowly. So in the early days of Lyme, there were two different views. There was the view that you just treated low dose forever for a long, long time. And maybe it would be 8 years if you extrapolated that you're going to treat over 700 replication cycles. And that way, you don't let that persister eat, in a sense, you suppress it. Or doing this very high dose to get MIC and BC adequately for an adequate number of hours to eradicate. And I wondered, do we know anything from our studies about that?
I think I would probably favor the latter. And the reason being is that Borrelia spirochetes can survive for, long periods of time in an unfed tick and still be infectious once they get a blood meal. I was astounded when I learned that the relapsing fever spirochete, Borrelia turcata, was transmitted to non-human primates. This was years back. After having starved the ticks for 5 years, they still transmitted into non-human primates and cause relapsing fever. And so I think these pathogens are extremely hardy. They can enter this dormant phase for a very long period of time. We don't necessarily know how long Borrelia burgdorferi can remain dormant in humans and in the presence of a low dose of antibiotic. The pulse dosing, what's been done in vitro, has been variable. It depends on what antibiotic you test. I think, Kim Lewis had some success with amoxicillin, and we didn't have any success with doxycycline. So this is why I probably favor combination antibiotics at a high dose. I think if you're going to wipe them out, you need to have multiple different mechanisms, something that targets the cell wall, combined with something that targets translation, combined with something that may, interfere with an enzyme. And that's the approach that we've taken with some of our research, and we've had some positive outcomes. And so I think it would be ideal, Ken can speak to this, but for a clinician to have a 30-day treatment regimen versus a year-long treatment regimen that's actually effective and doesn't really disrupt the gut microbiota over a period of time. That's another consideration, because how much you damage the gut can really affect the patient's health. So, and I know that there are ways around that, but this is our goal, to come up with something that is a shorter term and that can eradicate the spirochetes.
That would be grand. I mean, a lot of us clinicians, we've explored the outer limits of duration of treatment with antibiotics, and maybe the outer limits of the dosages. And at least with the regimens that have been available, a lot of us have concluded we are not getting rid of this infection, and our treatment is suppressive. It's not a great situation, but what's worse is if you don't treat at all, and then people are allowed to deteriorate it and suffer. So we need better tools. As far as intravenous antibiotic therapy, certainly, use my share of intravenous antibiotics, but there's a lot of risk that's associated with that. We just got a notice from one of our hospitals what a problem this Candida auris is. It's a very, very resistant fungus. So, like, goodness gracious, that's the last thing you want, is to have an indwelling line and have a patient get infected with something like that. So, yeah, I mean, it's one thing to have an IV line in for a couple of weeks at most, shorter the better, or even better, have agents that do not require use of intravenous approach at all.
And I would love a 30-day treatment. I've also wondered, there are other models for this. For instance, in small intestinal bacterial overgrowth, which is an overgrowth of bacteria in the upper intestine, the treatment is quite effective that's on the market, but you often have to pulse it 3 or 4 times before you really settle down. And I have wondered, given that maybe the lymph node is destroyed in some of my patients, but that doesn't mean it can't repair, right? We repair everything. So if you pulse in, kill the organism, come out, let the tissue repair itself, could then you get your seroconversion? Because I certainly do have patients who do not seem able to make IgG to Borrelia. And they do in general seem to stay sick longer. In terms of treatment, any comments about Herxheimer's, either in our primate cousins or in your disulfiram or other group? What is a Herxheimer jarish, and to what degree do we look at that as a guide to treatment or a guide that we're on the right track?
Well, it was originally described like 1900 or something like that by these two European physicians, Jarisch and Herxheimer. And I guess they weren't using antibiotics because antibiotics hadn't been invented yet. They were probably using heavy metals or something. And they noted that the patients with syphilis, when they first got treated, they would get very, very sick. And in fact, the Herxheimers could be so severe that they were fatal. And presumably that's from the breakdown of the organism, the release of mediators of inflammation, chemokines, cytokines, and whatnot. And so many clinicians use that somewhat as an indicator that the patient actually harbors a spirochetal infection. I think there had been some sense that, oh, you're a Herxing, that's great, you're killing spirochetes, but in the meantime, the patients are thoroughly miserable. And from my own perspective, I don't think it's desirable to emphasize or seek to have a huge Herxheimer's. Yes, it can be useful as an indication of what you're dealing with, but I think it can be destructive to persist when patients are having such enduring such misery. So I usually try to minimize it, or have patients sometimes back off on the treatment, suspend temporarily, and then resume at a lower dose, perhaps. So I mean, I don't think it's either necessary or desirable to have huge Jarisch-Herxheimer type effects. I don't know if you see that. Is there any correlate of that in the rhesus monkey, Monica?
We haven't seen that. I think because we treat, we treat a disseminated infection, and maybe they just don't have a heavy load of spirochetes. I wonder sometimes in human patients if it's related to other co-infections as well that might be aiding in this generation of the Herxheimer reaction. It's not my specialty, but the analogy to relapsing fever again is that in relapsing fever, there have been cases where mothers have been treated and had Herxheimer reactions that were fatal to them or their child. And so you do have to be careful, because when these spirochetes are dying, they're releasing things that cause inflammation, and you can end up with something analogous to the cytokine storm that you have in COVID, and it can be very dangerous. If I can back up to the treatment and the eradication of infection,
yes.
One point I think with having a 30-day oral treatment that we know eradicates infection, if we can get to that point, then all the other symptomatology that patients have, we can be more certain that it's not the result of a persistent infection, and we can use the appropriate tools. I say we, but you, the clinicians, can use the appropriate tools to deal with the inflammation or the arthritis, the autoimmunity, whatever might be causing the rest of the symptomatology. Because right now we don't know if a patient has chronic illness, if it's genuinely a result of persistent infection. And until we have something that we know can wipe out the spirochetes, we'll still be wondering.
Yeah, I think we know in some cases that there is continued infection, but I suppose the argument is, does the continued infection cause the symptoms? There's always that argument underneath there. I also have felt and seen in some of my cases, but these need to be studied, is, for instance, I often your neuroimmune antibodies and chronic Lyme patients with neuropsych or neurocognitive problems, and they're often high. If they are effectively treated, they drop. Sometimes they drop, below the normal. And so I've always wondered when we say autoimmunity, because we see this activated immune response that keeps on going, if it is persistent, sneaky, as you said earlier, stealth infection that kind of comes out, replicates, maybe the peptidoglycan is out there, and then before the immune system fully responds and goes through its cycle, or maybe manipulated, it never gets to that Th2 type. It's gone, and then it happens again and again. And so it looks autoimmune, but it's really infection driven. I mean, I would love to have a test that tells me if Lyme is there, or Bartonella is there, or Babesia is there, which is the other side of this is where is our testing getting us?
This dichotomy between infection versus autoimmunity or inflammation, like it's my impression that they can both be operative. They're not mutually exclusive at all. And of course, yes, if we had truly guaranteed eradicative therapy, then we could deal with the post. You could be sure that it was a post-infectious or inflammatory process. But right now, given the tools that we have, there's just a lot of uncertainty.
I also think that the immune system, as you said, is very involved in to go back to the Herxheimer reaction. And I think it is a concern. I think you can do damage. I think very simply, I think you kill the bug, it's in the motor neuron, for instance. All of a sudden, the immune system knows it's there and will attack. That's not a good place to have inflammation. So I've always thought the same about the Herxheimer, that you don't need to feel worse to get better. Before finishing, I want to just understand tolerance versus resistance. And yes, I'm looking at Dr. Embers here.
So basically, we don't know that Borrelia has any mechanisms that other bacteria present or possess. Some bacteria have things like efflux pumps where they can pump out the antibiotics, that get into the cell. And so when you treat a bacterium, a culture of bacteria with an antibiotic, if there are resistant bacteria in that culture, they're going to grow just fine in the presence of the antibiotic. They will grow like they normally grow. Tolerant bacteria will not grow. They'll just stay stagnant, but they won't die.
Mm-hmm.
Remove the antibiotic, then they'll slowly regrow. So they're basically adapting to a slow growing dormant phenotype that allows them to live in the presence of the antibiotic without growing. Does that make sense?
Makes perfect sense, yeah. In terms of co-infection, we know that ticks carry a lot of bugs, not all of which can infect humans or primates probably, but many of which can. And to what degree, Dr. Embers, are you looking at co-infection? I mean, one of my pet peeves in the area is that there's so little out there on co-infection. I know that if people get Babesia and Lyme, that they have a harder time getting over either infection. The same with anaplasma in one or two papers. And there are case reports of I know in Europe there's a chlamydia Lyme, there's a toxo Lyme, but there's very little serious investigation into what changes in the immune response. You know, there was a case at Duke of Rocky Mountain spotted fever, and the patient also had ehrlichiosis. He came in somnolent, and they found the Rocky Mountain spotted fever under the scope by staining, but he never mounted an antibody to that. Whereas to the Ehrlichia schiffensis, he did a fourfold change and came down. So I'm curious if that's looked at all, or what clinically you do can to assess COs.
Well, sorry.
I talked to you both. That was different.
So with respect to animal models of a co-infection, there are very few studies. There's some work out of the Parveen lab showing that Lyme disease, the inflammation of Lyme disease is more pronounced in mice that are co-infected with Babesia. And we've been very interested in looking at co-infection in the non-human primate model. I think if you approach co-infection with the perception or the knowledge that Borrelia can be immunosuppressive, directly immunosuppressive, then what might be a low-level infection of self-limiting infection, Bartonella, all of a sudden becomes a lot worse. And I think, we need to test for co-infections first off. Anaplasma and Babesia are reasonably treatable, but Bartonella is a problem because it's not easily treatable. So if a patient is co-infected with Bartonella and Borrelia, that treatment becomes a lot more difficult. The parallels between these two pathogens are, I'm learning, very striking. So I think we need to be very worried about what Bartonella can do, especially in the context of a Lyme disease patient.
And both very complex, multi-system, resistant, a lot of overlap of symptoms. So it's a challenge for sure. And I think the same thing, just like we need, improved methods of treatment for Lyme, we need improved methods of treatment for Bartonellosis. Of course, there can be some overlap, but I think a lot of the clinicians are also concluding that, we definitely need improved methods of treatment. It was Dr. Zhang's work that raised the question of whether clotrimazole, in the test tube seemed to have activity in methylene blue. Do you have any experience or thoughts about that? Or have those been applied in animal models?
There aren't really any good animal models of Bartonella. We're working on that. Yeah, the mouse model has been very problematic. And so, co-infection is something that we're pursuing in mice to see if we can get better penetrance and persistence of Bartonella if mice are co-infected with Borrelia. The tick transmission, it's still technically a question, but Bartonella can use so many different vectors to transmit that there's no reason that ticks are not likely one of them.
The proof has been hard to find though, right? Still contested.
I think it's hard to prove it, period, as I recall. And because there's so many other vectors, you'd have to have a very controlled situation. There's no dog model of Bartonellosis?
Dogs have been used for studying Bartonella, but they're not an ideal model. Actually, the cat's probably the best bet for a good model because Bartonella is known to persist long-term in cats, and in dogs, it can cause disease, but doesn't necessarily persist as long, and they're not as bacteremic as cats are.
Uh-huh. Interesting. What, let me check my notes to make sure. I'm curious from each of you if well, I will talk a bit about this. One of the pieces of information that I sometimes get when I am treating someone long-term with doxy or azith is that it's possible that I'm just using that antibiotic as an anti-inflammatory. And I mentioned earlier that nonetheless we apply anti-inflammatories and they don't work and the antibiotic does. But I'm curious that there's also a fair amount of antibacterial activity in drugs we don't classically think of having that. Some of your tricyclic antidepressants have that. Some of your H2 blockers, the azole drugs. And I wonder if you've thought about that, Ken, in treating people, if you've ever wondered if the antibacterial property, for instance, the tricyclics disabled some of the pumps in the bacteria. If you've ever wondered if, in a sense, that's your second antibiotic, because I have.
I haven't really systematically looked at it. I mean, I'm aware of that that article from Europe, Chart, that made the claim that fluconazole had antiviral properties. I know some of our colleagues have incorporated that into their treatment regimens. But maybe you should tell us what your thoughts and experiences. Vincent, obviously you've given thought to it, and maybe put it into practice.
So the what I've mentioned is that I do use fluconazole, thinking that I am most likely addressing a persister of Lyme, and also controlling yeast overgrowth. So, so I will use it. It has a QT interval problem, so I don't always mix it with a lot of things. I've mostly used it in working with psychiatrists and observing which drugs they choose that seem to work in the patient. The other comment I'll make is in my patients with seizures, apparently secondary to Lyme, the benzos work much better than my anti-seizure medications. And those are not necessarily antibiotics, but they are mast cell stabilizers. So, so I do, and disulfiram is maybe an example of this. I start realizing it's important that we look not at the class of drug, but at the mechanism of the drug and understand it with relationship to our bugs, our drugs for our bugs.
I think disulfiram has a lot of immune modulating properties too, not just but we're presuming antimicrobial properties. So it could be, multiple mechanisms that it's having action.
And maybe that is what we will, as I think Monica said earlier, that's what we'll come to, is coming to all those mechanisms. So I'm curious going forward, Monica, what would you like to see looked at? What would you die to get going in your lab? What do you see, solving? And same question to Dr. Lieger.
I can't think of just one thing, but of course we want that 30-day course of antibiotic that will eradicate the infection. I'm also very interested in the role of both Borrelia and Bartonella in neuropsychiatric disease and illness that may or may not be undiagnosed, or may or may not be diagnosed. And so what percentage of Alzheimer's patients, multiple sclerosis patients, ALS patients may have an underlying Bartonella or Borrelia infection that is inducing their disease. I think the microbial etiology of neurological disease, tick-borne, tick-transmitted pathogens in neurological disease is something that's fascinating me, and something that I'd like to look more deeply into.
If you look at all of the body of work that's been done on treatment trials for Lyme disease, for example, it's very paltry. There's just very few studies over the past 40 years. And I'm fond of making this comment that, for HIV, 2 to 3 billion a year, every year, has been dedicated over the past 40 years, and look at all the progress that's been made, as opposed to maybe 20, 25 million a year for Lyme. And a lot of that has been not acknowledging the problem of persistence. So once you acknowledge a problem, you have a chance of solving it. So I think there needs to be a lot more attention and support, both from private sources, private public sources, and public sources from the government to support proper trials and investigating new agents. There's a lot of work that needs to be done. I've said that in other venues, and it remains true.
And I think the neurodegenerative aspect is needs to be looked at, not just for tick-borne disease, but in general infections. Whether you go toxo or and given the predicted incidence of Alzheimer's, for instance, in the world. I read one comment that said there won't be enough people to take care of the people who have the dementia. And I certainly have at times thought it must all be an infection, and we aren't smart enough yet to figure it out. So I think that doing the careful, look at these wonderful primates will help. There have been some biopsies, et cetera, in humans, but we can't run around doing that a lot.
Well, there's again the autopsy studies that are being done both through Columbia and also through Bay Area Lyme. I think they're working together. That's another source of knowledge,
And I could backtrack to clinically. I know you have the pathology paper on your patient. In terms of where Lyme itself can and does go, and I know you said it's different in different primates, and maybe it's a matter of their genetics, and maybe it can't be a matter of the bug because you're using the same bug. But is there any tissue that Borrelia does not go to?
That's an interesting question. We haven't taken everything to look. So we would have to take everything in order to really answer that question. You've seen it in ocular disease, in the skin, in the we haven't really looked at like ovaries.
Yeah, I was just thinking endocrine.
Yeah, I think eventually we might get some hysterectomy samples for patients to look for that. Knee replacement, surgical discards, to look and interrogate them for Borrelia. We are involved in the analysis of autopsy specimens that Ken just alluded to, and we've gotten some interesting data already. So
I'm glad it's being done. Well, thank you very, very much. And I hope to hear from you about the next paper and to learn more as disulfiram unfolds.
Absolutely.
All right.
Again, thanks for inviting us to speak.
Well,
Have this-
thank you for speaking.
Thank you. Yeah.
Have this conversation.
All right.